Hello, and welcome to Talking About Regeneration, the podcast that's all about stem cell and regenerative medicine and some other stuff too. I'm Kevin McCormack, Communications Director at CIRM. We're California's stem cell agency, and we have $5.5 billion to change the world. And this podcast will introduce you to some of the people who are going to help us do just that. Enjoy the show. Our guests today are three scientists from the Gladstone Institutes here in San Francisco. The Gladstone, for those who don't know, is an independent, nonprofit biomedical research organization whose focus is to better understand, prevent, treat, and cure conditions such as heart failure, HIV/AIDS, and Alzheimer's disease. Dr. Yadong Wang is the senior investigator and director of the Center for Translational Advancement at Gladstone Institutes. Dr. Nicole Koutsodendris is a graduate student in Dr. Huang's lab and a co-author on a recent paper in the journal Nature Aging. We're going to be talking a lot more about that on the podcast. And Dr. Lana Juladeva is a postdoctoral scholar in the lab of Dr. Deepak Srivastava. And for those who don't know Dr. Srivastava, and it sounds like I don't because I can't pronounce his name, he's something of a rock star in the world of regenerative medicine. So Lana being in his lab is obviously big kudos for her and for us for having her join us today. So doctors, we're delighted to welcome you all to our podcast talking about regeneration. And thanks for taking the time out of your labs to join us.
Thank you for having us.
Thank you, Kevin. So it's a great pleasure to talk with you about our research.
Great. Actually, Dr. Yadong, if we could start with you. You and Dr. Nicole, I'm just going to use first names here because it's easier and I'm less likely to mangle them, recently had a paper in Nature Aging that showed how a drug that's been around for about 30 years might actually be useful in helping combat Alzheimer's. Talk to us a bit about that work, if you could.
Yeah, so this is a recent work about a drug repurposing. The purpose of the drug repurposing is to find some already approved drugs which can be on the market for many years, whether that drug can be used for a different disease, in this case, for treating or preventing Alzheimer's disease. That was the goal. Starting around 5 or 6 years ago, we wondered for so many years, the drug development for Alzheimer's disease, as you know, have been very difficult. A lot of money spent on this area, but the progress were not that great. And so we wonder at that time, in order to develop new drugs that can take about 12 years on average, spend billions of dollars on new drug, whether we can significantly shorten this development time and also lower the cost by repurposing some existing drugs. That was the goal when I started this Gladstone Center of Translation and Advancement. The major goal is to develop drug repurposing work. So at that time, we think if you look at this Alzheimer's disease, it sounds like a single disease, but if you really look at the molecular mechanism underlying this disease, you will realize it might not be a single disease. It may be a combination of different subgroup of this patient. Each subgroup may have their own underlying mechanism that lead to this disease, although on surface, they all have dementia. So this is tells us probably we need some precision medicine to really deal with this complicated aging-related disease. So what we decide to do for the first step to do this, to apply this precision medicine by analyzing this ApoE4 genotyping among those all AD patients. As you know, ApoE4 has been identified as the major genetic risk factor for Alzheimer's disease. Anybody carrying one copy E4 accounts for about 20-25% in normal population, but in Alzheimer's patient, it accounts for up to 70 or 75% AD population. So we split this Alzheimer's disease patient into E4 carriers and non-carriers, and then we look at their phenotype. Basically, we not only look at their pathology, their clinic phenotype, but look into their very basic change in their cells. Basically, among those little about over 20,000 genes everyone has, what are those general changes of those genes between the E4 carriers AD and the E3 AD to see whether they are overlap, whether they are different. Strikingly, actually, E4 AD only share about 6% altered genes of those over 2,000 genes with this E3 AD. That tells us again, E4 AD might not be same as this E3 AD in terms of this gene expression level. So we start to search this known drug database covering about 1,300 drugs to see which drug can actually flip or reverse those Alzheimer's disease related gene expression profile specific for this E4. And then we rank those 1,300 genes and we look at this top 5 genes. So that was the time we identified this water pill, so-called, this bumetanide among the top 5 candidate drugs. And then we test this drug in animal models of AD and also in some real world human data. And also importantly, use this human iPS cell derived neuron model to test this. After this validation, now we realize this drug might work at least in animal models and in the real world data database. And then probably the next step will be moving to clinical trial to test this drug.
So this is the story. Straightforward. You make it sound really straightforward, but I'm guessing it took years and years. And Nicole, how many different drugs and medications did you look through before you found one that seemed to kind of fit the profile that you were looking for?
Yeah, so I think the strength of the study is, using this database of, already FDA approved drugs. So I think that helped narrow down the list. So the database in total had 1,300 drugs in it. And then it was, Yadong and Ali and the other co-authors, methodically looked at these gene expression changes, but also we're looking to see, for certain factors, like do these drugs easily cross the blood-brain barrier? Because, further down the line in clinical trials, that would make things a lot easier if the drug was able to do that. And also, making sure that there's a long-standing records of this drug being safe in humans. So that's ultimately what helped us narrow down the list from 1,300 drugs to bumetanide.
So Yadong, what were the results from these various tests that you tested this drug on?
So the major outcome after we identify this top 5 drugs, we look at literature, identify bumetanide might be the best we can try. We went on to test this bumetanide treatment in 2 mouse models of Alzheimer's disease. One is the mice express human ApoE4 in their cells. And then these E4 mice will develop Alzheimer's disease like learning memory deficit at age about 15 months. And then after we treat these E4 mice for bumetanide, with bumetanide for about 8 weeks, and we see this treatment completely rescue this memory deficit, learning deficit, at the same time also rescue this abnormal brain activity if you do the electrophysiology and recording, which underlying this learning memory deficit, that also was rescued. And then we went on to even more complicated mouse models in which there are both ApoE4 expression at the same time accumulation of amyloid beta in this mouse brain, which is one of the major pathological hallmark in human AD brains. We look in this second model, again, in this case, 3 months of bumetanide treatment also completely rescued this abnormal brain activity associated with Alzheimer's disease and also lower 50% the amyloid load in those mouse brains. And then after we complete this study, we went on to further test whether this also works in human cells by basically try to bridge the gap between the mouse work and the human implications there. So we use this iPSC-derived human neurons carrying the same E4 genes. And then after we treat this E4 human neurons with bumetanide, we see this ApoE44 associated gene changes that we observe in human brains. Many of them were reversed back to normal status by bumetanide treatment. At the last step, further towards human use, and we look into this so-called real world, this electronic health record database, two databases. One is from UCSF, one is from Mount Sinai Hospital. And then both databases cover over 5 million patients. We filled this database by pulling out this anybody who took this bumetanide for many years, and they are older than 65 years, which is usually the time AD start to occur in humans. And then compare with those people who did not take this drug. We found roughly 35% to 75% reduction of AD, Alzheimer's disease prevalence in both databases. So that further suggesting this drug might be protective for those humans to develop Alzheimer's disease. So that's the major outcomes after we identify the drug and test in mice and in human iPS cell neurons, and then in the real world data.
That's amazing. I mean, not just that it works in mice, but also in the iPSC cells, the human model, but then also to see that evidence in the real world. Mm-hmm. So what does this mean in terms of being able to move this forward into, say, a clinical trial? As you talked about, it's already been shown to be safe in people over many, many years. And so how long would it take to kind of get this into a clinical trial to see if it does have protective effects for people who are at risk of Alzheimer's?
Yes, so now clearly the next step is go to the real clinical trial. As you said, this drug has been used in the clinic for about 40 years. Its safety profile has been well established. It's a so-called water pill, basically get rid of extra water, fluid from our body under some condition, including hypertension, heart failure. So, but because it's a strong diuretic drug, so that can cause dehydration if anyone take too many or too frequent. So we do need to go test in the Alzheimer's disease patients about this drug. So I hope, I'm now currently working with multiple medical centers, try to initiate this clinical trial. I hope we can start middle next year for a real clinical trial. Of course, we'll start with those Alzheimer's patients carrying this E4 mutation, and then test whether this drug will work. After that, we probably can expand more broadly for more Alzheimer's patients on that.
That's exciting stuff, and moving along fast. But do you worry sometimes that when you have a finding like this that looks so promising, that maybe some people might start going out and trying to get prescribed bumetanide for themselves, regardless of whether they actually need it?
Yeah, you're right. That was the reason we have been putting in all these notice in all different places. So we try to, say, don't use this drug until this real clinical trial approve it is effective in human patient and with a very safe profile of that. So we are actually cautious about using this drug off-labelly to any patient at this stage.
Right. Nicole, as a young researcher, it's quite a coup to get in on a study like this that has such profound implications potentially.
Yeah, it was incredible to be part of such a study that I thought was very extensive. And I especially liked how, at different levels we were able to test bumetanide. I think using a combination of mouse models and human iPSC-derived neurons, and also, the human database was incredible to kind of show proof on many different levels that bumetanide is effective for, preventing certain features of Alzheimer's. So yeah, I think it was a really good way to see how good science is done. And it's good to see that, early on in my graduate career.
Oh, yeah. Lana, if we could come to you now, you're working in very different areas. So talk to us a little bit about the work that you're doing.
Sure. I just wanted to comment as I was, listening to both Yudong and Nicole reflect upon their work. It's kind of like one of the perfect examples, I guess, from basic science to clinical translation to then back to the bench, and all of these three things combining, coming together in such a perfect relationship. I mean, it really speaks to what we do here at Gladstone. And my work is a little bit different, but I'm taking a slightly different approach in tackling neurodegenerative disease. But I still use, the stem cell technology that we have here in the institutes and the power of our stem cell core. But my primary model is using human-induced pluripotent stem cells to engineer a particular type of neuron. They're called spinal interneurons. And in particular, I'm focused on studying the injured spinal cord and how we can enhance recovery after, something as traumatic as a spinal cord injury. So the vision and the technology, they still align. And I'm also interested in, taking and thinking and doing my bench top research, but other activities, both through Gladstone as well as through conferences and seminars and symposia, allow us to look at and consider the translational perspectives in our work.
And that's such an important part, because I think for people who don't know, basic or discovery research is pretty much the starting point for all of this. And then if it succeeds, it moves into the translational phase. And obviously, if that succeeds, then hopefully it then moves into the clinical trial phase, where it's being tested in people. So it must be really satisfying to be able to see work like this that starts out as someone has an idea and someone asks the question, I wonder why that's the case. And then to see it progress through lab work, through ingenuity and different ways into a more promising phase.
Absolutely, yes. And I think it starts by including, people like myself, trainees. So people like myself and Nicole at the early stage where we gain an appreciation of what we are doing here at the bench matters. Even if it's not going to go directly into a person next week, everything that we're studying, all of the little, tinkering and assays that we're doing, it's all contributing to the big picture. And having that vision and being aligned with it from early on as a trainee is incredibly important for that success that you mentioned.
Right. And also I think trainees, postdocs they maybe come in a little naive and may ask questions that a more seasoned researcher may think they know the answer to. And you might just say, well, what about this? And that opens up a whole other conversation.
Absolutely. And that's where mentorship is a huge part at Gladstone. So all of our mentors are incredibly sensitive to what may sound like a crazy idea coming from a graduate student or a postdoc, but I haven't met anyone who shut an idea down. They just, we have that ability, we have that freedom, and just, okay, go ahead and explore it and come back to me, tell me what happened last week. And that's where the conversation really starts to get going.
So, if we could come back to you and talk to you about your feelings about that idea, about having obviously at Gladstone, you can attract some of the smartest, brightest postdocs around. But how helpful is it to have that fresh perspective?
Oh, that's very important. Like Lana just mentioned. So I can tell you the starting of this this study. The first author in this paper, unfortunately, she cannot join today. She gradually left Gladstone, Alice Tobey, when she was a graduate student in the lab. So I was studying, ApoE4 and Alzheimer's disease. She was assigned to a project. It's completely different from finally she worked on. And then into her thesis work about a year. And then I remember one day we sit in my office, just decide what will be her next step. And then I mentioned about this drug repurposing, and she started to be very interested. And then she further read many literature. She came back saying, I think this is a really great, area. She is interested. And she actually decided, she asked me whether it'd be okay for her actually switch her thesis work into a very different drug repurposing field. And then because of her strong interest, her knowledge about computational analysis, I thought that's great to fit to her goals or interests. And then she switched from our original project to this drug repurposing project. And then since then, she led this project because my lab was not built on this kind of a drug repurposing or computational analysis. She actually took the challenge as a trainee and a graduate student, and then really lead this project by reading the literature. And of course, we add Marina Sirota, another faculty from UCSF, to also co-mentor her because she has this computational background. And then she actually developed from, zero. We don't have any of this experience in our lab doing this kind of work. And she built from zero ground and then gradually to generate more and more data. And finally, we got this very fruitful, outcome finally. So this is, again, says, how important is young trainees, including graduate students, their contribution, just, so much there.
I know it teaches you to be careful about what you talk about around them because they'll get great ideas and then they'll go off and do something else.
Right.
So
right.
Nicole, do you ever feel that you're kind of at Gladstone, because it's more kind of on the earlier stage, that as researchers, you're kind of not looked down on, but overlooked? It's not as glamorous as some of the other kind of the research fields.
Yeah, I'd say that it kind of depends on what we're talking about. So I'd say that amongst like the research community and clinicians, that they kind of understand the value of basic research. But I would say, like, I agree with your point that maybe in a more public health, like general public perspective, that, sometimes the clinical trials get a lot more attention than the basic science research, whether that's because, more general news companies aren't not necessarily like publishing articles about the basic science research. They mostly like to, publish articles about anything that involves like therapeutics or clinical trials. So I think it is, like, on one hand, an issue of our research not necessarily reaching the general public. And it might also be an issue that they might not necessarily understand the implications of basic research and how that then relates to, drugs that we find that can be used in clinical trials.
Right. So Lana, how would you make the argument to the public if people ask you what you do that highlights just how important this kind of research is in paving the way for all the potential future therapies?
I would say, thinking about even the vaccine against COVID and how quickly that was developed, I would say, take a look at how much research went in, how many decades of research went into the mRNA technology. And only then do you get a sense of appreciation that, oh man, yeah, this research has been last 30, 40 years, and this technology has been around for the last several decades. And that basic research, that decades worth of work, is what enabled us to accelerate the translational pathway for something as important as tackling this disease. And the same thing goes for every other disease. We don't know how important our little discoveries are and how really big they are. And so I really do think that the best way, I guess, for the public to understand the value for basic science research is talk to us. Talk to the basic researcher, talk to the trainees that are doing their PhDs and what that means. Talk to the postdocs who work in laboratories all around, San Francisco and across the Bay Area and, beyond. Mm-hmm. I think that, that is one of the best ways to really understand what goes into it and what the vision is. and really see how we envision all of these discoveries then giving back to the public.
Right. And
I want
if we could end with you just talking about, you've been doing this for 30 years. You're the poster child for sticking with kind of basic research until finally you're able to kind of pass something out of it and produce something that might be a game changer. I mean, it must be very gratifying for you after all those years.
Yes, that's true. And I also want to emphasize this why this basic research is so important. If you look at this outcome, yes, we finally probably figure out one potential drug to treat Alzheimer's disease, but it's all depend on many years, like you said, 20, 30 years of basic research. So I mentioned for many years, drug development against Alzheimer's disease has been focused on the known pathology, tau pathology, A beta pathology. But our study highlighted, it might work, but we need to go step back, look at the basic science. What we did is instead of look at this individual pathology, and then we say, okay, let's look all those 22,000 gene expression. Those are really basic. Every protein, in order to function, they need, get the translation from their gene. And then we look at this gene expression, not thinking about any known hypothesis, just see what are the gene changes. That gene change profile guide us to see which drug might work that capable of, reversing those altered genes. That's exactly why we see this potential drug, because we apply this basic science about the gene expression change toward this disease altered gene changes, find some drug finally to reverse those gene change that might work. So this, again, like I said, really highlight how important those basic research can actually lead us to a potential fruitful, a product potentially for treatment.
Yeah, and when you think how many drugs and new approaches to Alzheimer's over the years have failed, or with the case of Aduhelm, not really shown huge benefits, it would be wonderfully ironic if a drug that had been around for that many years actually had more benefit and could help far more people than some of those other ones, with a minimum of the costs that we were talking about, the billions of dollars it might take to develop.
Yeah, I completely agree. So if you look at this water pill, if finally it would work, it costs about $25 a month.
Wow.
So if it works, that'll be great for patient.
Absolutely. Well, that's all we have time for. So I would like to thank our guests, Dr. Yadong Huang, Dr. Nicole Kutzadendris, and Dr. Lana Juladeva, for joining us and sharing their research with us. Thank you all. It's been fascinating.
Thank you.
Yes, thank you for having us.
Thank you for having us. Yes.
And I'd like to thank you at home, in the car, the gym, the shower, or wherever you listen to podcasts for joining us. And we'll look forward to seeing you next time on Talking About Regeneration.